Hello, and thanks for showing interest in my independent study from fall of 2020. Um, we're going to take some time to talk about some data explorations that I did to explore levels of gene expression for a couple of genes and how those might relate to the survival of cancer patients with a few different cancer types. So what am I talking about? Well, what we're looking for are tools to help us find out what the probability of survival at time X might be for a patient to help make treatment decisions, life planning decisions, etc. These tools could be macro, like tumor size or metastatic status, or they could be molecular. For example, the gene HER2 is a growth factor for embryos, but when expressed in tumor cells, can lead to lower survival times. So, how might we go about choosing a gene like HER2 to study? Well, we might look to see if the gene has an effect on cell growth as a logical place to start. There's a phenomenon in cancer cells called the Warburg Effect, where cancer cells in their constant replication require a lot of energy, and they seem to prefer fresh glucose over any other sort of energy pathway for energy. So their demand for glucose is much higher than for many non-cancer cells. So then perhaps investigating genes that produce products that help cells consume glucose may provide insight into the cancer cell's ability to process glucose and grow and replicate. The thing is that cells require glucose transport factors, or GLUTs, to facilitate glucose diffusion into the cell. There is a family of them, but the most common is GLUT1, and in some, GLUT2 in cells like liver cells. All right, so knowing that if we feed a cancer cell more glucose, it may be more able to replicate, we have our research question. Do more GLUTs can more GLUTs be associated with a worse prognosis? So we'll attempt to get to an answer to that question with data provided through next generation sequencing technologies, technologies that allow us to quantitatively measure the amount of all of the mRNA that is like the blueprints for proteins in a sample. The internet has given us portals to distribute these. Ours came from the CBio portal, which distributes samples collected from the Cancer Genome Atlas. And we're going to look at the particular expression of the genes SLC2A1 and SLC2A2 in several different cancer types. These code for the two most common GLUTs, GLUT1 and GLUT2, respectively. Okay, so now that we've chosen our proteins of interest, GLUT1 and 2, and therefore our gene of interest, SLC2A1 and SLC2A2, how do we choose which cancer types to study? To have samples to study. Well, we see here an image that shows many of the cancer types that have studies on CBio portal. Shirley Pagan here, my research compatriot over the term, has ranked them by the amplification mutation percentage. That's by copy number. And the thought that we had in using this ranking to determine specific cancers in which to study was that increased rates of expression of SLC2A2, that is increased amounts of GLUT2 or GLUT1 for that matter, would likely be the product of the copy number mutation, the amplification, presumed amplification mutation. So you'll see that the cervical squamous cell carcinoma, for example, one of the cancer types that we selected for this analysis was high in the list. So in addition to the cervical carcinoma, We also selected the hepatocellular carcinoma, pancreatic adenocarcinoma, and kidney renal clear cell carcinoma. This is because the liver tissue relies on GLUT2 as its primary glucose transporter. GLUT2 expression in liver cells was also used in a research paper that served as the model paper for my own work. And so crunching the same set of data as Kim et al., the model paper studying the hepatocellular carcinoma, was a way of validating my methods just by seeing if my same program would deliver similar results. So the survival that we took from the studies were a patient ID and a sample ID, the patient's survival status, the patient's survival time, and the mRNA expression data for the genes of interest reported as deviations from the mean. These expression levels were divided into groups and then plotted against one another with a statistical tool called the Kaplan-Meier estimator, which is used to study survival. It's a survival analysis tool. So this is the type of chart used to report my findings. You'll see on the y-axis the survival probability, and on the x-axis the survival time in months. You'll also see that the probability drops at every time there is a death, and there are also small marks that represent censoring events. This is when someone leaves the study without dying, and so we don't use their leaving the study to change the survival probability, although it does shrink the remaining amount of the remaining pool of survivors, so it affects it does affect the data. You'll see multiple curves plotted on the same set of axes and a confidence, a p-value that just indicates our confidence that the curves are different. Actually quantifying whether or not expression levels are positively or negatively affecting survival outcomes is a finding that will need some additional analysis, but at least we can be confident that there is difference in the curves. So this is some of the statistical analysis that I performed. You'll see that increased SLC2A1 expression is associated with decreased survival in hepatocellular carcinoma. That is, greater amounts of SLC2A1 expression resulted in a worse prognosis, less likely to live as long. You'll see here that increased SLC2A2 in hepatocellular carcinoma conversely results in greater survival time for greater than median expression groups. These two findings were similar to findings made by Kim et al. in their the paper that I used as the model for my own research. You'll see here that increased SLC2A2 expression is associated with increased survival in kidney renal clear cell carcinoma. Increased SLC2A1 expression is not significantly associated with decreased survival in kidney renal clear cell carcinoma. And increased SLC2A1 expression may be associated with decreased survival in kidney renal clear cell carcinoma, especially at highest levels of expression. So here's a table of my findings. The hepatocellular carcinoma findings were from our model paper, but the other three cancer types were not investigated previously. Cervical squamous cell carcinoma and pancreatic adenocarcinoma did not show significant variance, but the kidney renal clear cell carcinoma appears to have significant variance in survival curves for expression of SLC2A1 and SLC2A2. So, in summary, we found significant variance in hepatocellular carcinoma in kidney renal clear cell carcinoma patients with varying degrees of GLUT1 expression and GLUT2 expression. In general, GLUT1 expression appears to deteriorate survival probabilities, and increased GLUT2 expression appears to improve survival probabilities. We did not find significant variance among cervical squamous cell carcinoma or pancreatic adenocarcinoma. Again, I just want to reiterate that there needs to be additional post hoc analysis to show definitively that the curves indicate those differences, but I have shown at least significant variance between those curves.